Hello, very good evening to the students who have joined. Can you let me know if my voice is audible to you and if you can see the video? In the chat box, please let me know if my voice is audible. Okay, thank you so much for confirming. Now, please let me know if you can see the presentation clearly. Let me know that if the PowerPoint presentation is clearly visible. Okay, thank you for confirming. Now, let us wait for a couple of minutes, then we will start today's session. Let us wait till it's 6 p.m. Then I will start today's session. Meanwhile, if you have any doubt or any query regarding the last class or the last class course content, you can let me know. We will start at 6 p.m. when most students will join, if other students join. So please wait till then. So it's almost 6 p.m. Let me just wait for a minute, then I will start the session. Okay, let me start by introducing myself. So, very good evening to the students who have joined. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. And today it is our week 6 live session. I hope my voice is audible to you, and I also hope that the PPT, the presentation is clearly visible to you. If not, please let me know in the chat box. So today it is our week 6 live session. So what we have done in our last live session, which was in the last Friday, so we basically have discussed about the DNA, about the nucleic acids. We have discussed the general basics of the DNA and nucleic acids. Also, we discussed about the structure of the DNA. We have discussed about the DNA sequencing method, just like we do in case of the polypeptide chain. We do the amino acid sequencing. We also can do the sequence of, we can identify the sequence of the nucleotides in case of a DNA molecule. So there are several methods like Sanger's method, there is a Maxim Gilbert method, and so on. So all these things and we have discussed in our previous class. So if you, I have already mentioned, if you have any doubt or any query, you can let me know in the chat box. Okay, so today it is our week 6 live session, and before starting the session, like every class, I would like to point out few things, that there will be a problem solving session every week till the course ends. Okay, this week's will be on every Friday from 8 to 6 p.m. If in any case the date and the time of the class is changed, means if the class is rescheduled, you will be notified via email. Okay, these sessions all are live on Google Meet platform. This is the link for this classes Google Meet. Okay, and this link will remain same throughout our session. You can write it down or save or copy this particular link to somewhere and use this particular link to join every class. Okay, this will be intact for our entire session. All I mean for upcoming classes also it will be same. So the important thing is, if any network disruption happen from my side, okay, the students, you should not leave the class. Okay, I will try to rejoin as soon as possible, but you should not leave the class. You have to wait for some time, and I will try to join as soon as possible. And since I am recording the session, so I am recording the session so that I can share this recording to you after the class. Okay, so that is why I have to mute your cameras and microphones so that I can record the session without any hassle. But the chat box is always open, so you can always post your doubts, your queries, anything you want in the chat box. I am monitoring the chat box every now and then, so if you can post your doubts or your queries and anything you want. If there is any issue with the audio or any issue with the power presentation, you can let me know through the chat box. Now, if after class any doubt persists, you can drop an email to me. I will share my email ID at the end of the session. Okay, in the chat box I will share my email ID. You can use that email ID to ask your questions. Also, you can let me know if you have any doubt or any query in the next class. Okay, and since I'm recording the session and so the classroom is always open, so if you have any network issue, you can rejoin, you can leave and rejoin at any time. Okay, so that won't be a problem. And this presentation and class recording, it will be shared to you. So where you have to find this presentation and class recording, I have already, I think, shown you in the last class also. So let me show you one more time. Is that you have to go to the SWAYAM portal. Okay, you have to go to the SWAYAM portal. In the SWAYAM portal, you will find there is a first thing is the announcements which are posted here. Also, the class reminders are also posted, like the okay. And like this, the class reminders is also posted here. So in the left panel, you will find there are weekly updates. So whatever is posted for a particular week's course content, it is posted here, and you will find that there is a problem solving session. There is a point which is written as problem solving session. You have to click on that and you have to go to the problem solving session recording. By going to that recording, you will be redirected to a page where you can download this particular PPT from a Google Drive. Okay, this all the PPTs is uploaded on a Google Drive, which looks like this. You can download the particular PPT, week's PPT from this Google Drive, and also the class the recording. It will be uploaded to the YouTube, and the YouTube link will be also shared in the same page, so you can watch the particular class recording if in case you have missed anything. Okay, so these are the things which I had to summarize before starting the session. So as I have mentioned, the chat box, not only we will use this chat box for our question answering purpose, also we will utilize the chat box for answering the each and every question. So what we do in each class, that in every live session I share with you around 20 questions. All the questions are MCQs, okay, which will be followed up by 4 different options, and one of the options is the correct answer. So after I show you a particular question, I will wait for 2 to 3 minutes. Okay, and meanwhile you have to identify which of the following will be the correct answer, and you have to let me know in the chat box that A, B, C or D, which of the following is the correct answer for a particular question. Answering during the session, during the live session or the practice sessions, problem solving sessions, so it won't have any kind of marking. So even if your answer is wrong, it does not matter, because I won't be marking and there won't be any kind of negative marking. But there are marks in your assignments which is posted in the SWAYAM portal, so you have to be very careful while answering those questions. Okay, but whereas during the class you can answer freely, whatever you think is the correct answer, you can let me know in the particular chat box. Okay, and after I check your answers, I will show you the correct answer, and for that particular question we will discuss in detail, so that if you are asked any kind of subjective questions, you can answer it easily. So today it is our week 6 live session, and today we will discuss the week 6 course content of this course of organic chemistry and biology and drug development. So what are the course contents? The contents are as follows. We have the synthesis of oligonucleotide, Then we have the central dogma, the DNA replication, transcription and translation. And we basically discussed about the central dogma today in detail, and along with some little bit I will discuss about the synthesis of the oligonucleotide, how it happens. So what is the central dogma? So basically central dogma is nothing but the flow of the genetic information from DNA to RNA to protein. Okay, so from DNA first happens the replication where the DNA strand follows another DNA strand. Then it happens, what happens is the your transcription. Okay, from where the DNA is converted to RNA, then translation where RNA is converted to protein, and the genetic information which it is passed through this DNA to RNA and protein, and it is unidirectional. Okay, it is not like the genetic information can reverse back from protein to RNA to DNA. It does not that does not happen. It happens only one way, from DNA to RNA to protein. So all the things related to this the genetic passing of the genetic information from DNA to RNA to protein. Okay, these via transcription, replication, transcription and translation. This is known as the central dogma of the molecular biology, which is a very crucial topic. Okay, in case of the very in our course, that is a very crucial topic. So today we will discuss the central dogma. We will discuss about replication, transcription and translation. Also we will discuss about the processes and the enzymes which are responsible for all these things. Okay, now let me start with the first question of today's session. So the question number 1, it says that which of the following statements regarding ribosome is true? Your options are: number A, ribosome of prokaryotes is larger than eukaryotes; number B, ribosome is made up of messenger RNAs and proteins; number C, the eukaryotic ribosome contains 50S and 30S subunits; and number D, the prokaryotic ribosome contains the 50S and 30S subunits. Okay. So the problem number 1, I will repeat. It says which of the following statements regarding ribosome is true? Your options are: number A, ribosome of prokaryotes is larger than eukaryotes; number B, ribosome is made up of messenger RNAs and proteins; number C, eukaryotic ribosome contains 50S and 30S subunits; and number D, prokaryotic ribosome contains 50S and 30S subunits. One of the following statements is true. You have to identify A, B, C or D, which of the following will be true. I will wait for 1 to 2 minutes, and then I will check the chat box and we will discuss about this thing in detail. Okay. Which of the following statements regarding ribosome is true? See, for answering during the class, even if your answer is wrong, it does not matter. But please try to answer whatever you think is the correct one. Let me know. Which of the following statements regarding ribosome is true? Let me check the chat box. Let me check if there is any answer given in the chat box for problem number 1. Okay, I got one answer which is saying that it is number C, which is correct for the first problem. Okay, let me check if number C is the correct answer or not. So the statement which is true regarding ribosome will be It is number D, which says that prokaryotic ribosome contains 50S and 30S subunits. Okay. So first, let us understand what are ribosomes. So basically, ribosomes are small, rounded, dense particles of ribonucleic protein. They occur either freely in cytoplasm and the matrix of the cytoplasm and the matrix of mitochondria and chloroplast, or remain attached with the membranes of the endoplasmic reticulum and nucleus. They are the sites for protein synthesis in both prokaryotic and eukaryotic cells. This is very important, okay, which you need to remember that ribosomes are basically the sites of the protein synthesis in case of both prokaryotic and eukaryotic cells. Okay, so if you are asked what is the site of protein synthesis and what organelle actually helps the protein or is responsible in or participates in protein synthesis, the answer will be ribosome. The cells in which active protein synthesis takes place, that is the pancreatic cells, hepatic parenchymal cells, plasma cells, thyroid cells, etc. The ribosomes remained attached with the membranes of RER. Okay, then cells with the synthesized specific proteins for the intracellular utilization and storage, like erythroblast, skin, and certain large number of free ribosomes in cytoplasm. Ribosomes are usually designed or designated according to their rates of sedimentation. The sedimentation coefficient is expressed in the Sedberg unit or S unit. S is related to the size and molecular weight of the ribosomal particles. So this is the prokaryotic ribosome. So basically the ribosomes have two different subunits. So first let us see the other statements of the number one statement, which says ribosome of prokaryotes is larger than eukaryotes. This is not true. Okay, obviously, because the ribosomes of the prokaryotes, it is smaller. The eukaryotes ribosomes are much larger in size than the ribosomes of prokaryotes. Ribosome is made of messenger RNA and protein. This is not true. Ribosomes are basically made of the rRNA, okay, not messenger or mRNA. And eukaryotic ribosome actually forms with the 60S and 40S subunit, okay. So in total, it is said to be the 70S, okay. The two subunits, one is 50S subunit and another is 30S subunit in case of the prokaryote ribosome. In case of the eukaryotic ribosome, the total it is known as to be the 80S, and the other subunits are the 60S subunit and 40S subunit. Okay, so that's why the statement number D is true, which says that prokaryotic ribosome contains 50S and 30S subunits. Okay, but eukaryotic ribosome, it contains 60 and 40S subunits respectively. Okay, so this was the correct answer, number D. Is there any doubt? If there is any doubt, please let me know. So whenever actually I show you a particular answer of a particular question, if you have any doubt or any query, you can let me know through the chat box then and there, okay, or you can ask your doubts in the at the last of the session also. Okay, so this was the first question. Let us go to the next question. So problem number 2, it says which of the following is the start codon and for which amino acid it codes for. Your options are number A, AUG and methionine, number B, UCU and serine, number C, UUU and phenylalanine, and number D, AAA and lysine. Which of the following is the start codon and for which amino acid it codes for? Or which amino acid it codes for? Your options are number A, AUG and methionine, number B, UCU and serine, number C, UUU and phenylalanine, and number D, AAA and lysine. Which of the following is a start codon and for which amino acid it codes for? Your options are number A, AUG and methionine, number B, UCU and serine, number C, UUU and phenylalanine, number D, AAA and lysine. Identify the start codon and the amino acid it codes for. I will wait for 2 minutes, then I will check the chat box. Okay, let me check because I think this is a much easier question, and I know all of you can answer it correctly. Okay, I got one answer which is saying it is number A. Let me check if number A is the correct answer or not. So the start codon and the amino acid it codes for will be very good. It is number A and methionine. Okay, so what is a codon? So basically codon is a 3 nucleotide sequence in mRNA where it actually denotes a particular amino acid. Okay, it codes for or it gives a signal for the particular in during the translation for incorporation of a particular protein. So there are several codons, and every codon has a specific amino acid for which it codes for. Okay, but the start codon always, if the start codon is universal and it is same irrespective of any cases, the start codon is the only one which is the AUG. Okay, so start codon is the first 3 nucleotide sequence in an mRNA transcript that signals a ribosome to begin the protein synthesis or translation, and it is also codes for the initial amino acid of the protein. The most common start codon is AUG, which translates for the amino acid methionine in eukaryotes and archaea, and for formyl methionine in case of prokaryotes and organelles. So how it works: the initiation of translation during the protein synthesis, a ribosome scans the mRNA molecule. Recognition: the ribosome recognizes the start codon, which is usually and most commonly it is AUG, as the start to begin a translating mRNA into protein. Amino acid incorporation: the special transfer RNA or tRNA binds the start codon and introduces the first amino acid, which is the methionine. Then protein elongation: the ribosome then continues to read the subsequent codons, adding more amino acids to the growing polypeptide chain. So what are the key facts? Key facts are like this. AUG is the start codon, the most frequent start codon in both prokaryote and eukaryote organism. Methionine, AUG codes for amino acid methionine, which is always the first, nearly always the first amino acid in protein. Alternative start codons, which is very extremely rare, but there are some alternative start codons like GTG can also exist, and they also initiate the translation. Location: the start codon is located at the beginning of the mRNA's protein coding region after the 5' untranslated region. Okay, so there is a 5' untranslated region. After that, this AUG start codon is there for coding the first amino acid, which is the methionine. Okay, so this was the first second question, and the answer which is correct, which is the AUG and methionine. Okay. So if there is any doubt regarding the codon or anything, you can let me know. Otherwise, I will move toward the next problem. Okay. So now let me go to the problem number 3. So problem number 3, it says that in prokaryotes, the first amino acid in polypeptide chain is, you have to fill in the blanks. Your options are number A methionine, number B N-methyl methionine, number C N-formyl methionine, and number D all of these. So in prokaryotes, what is the or which of the following is the first amino acid in the polypeptide chain? Methionine, N-methyl methionine, N-formyl methionine, or all of these? If you have paid attention to the answer for the last question, I think you can easily answer this question because I have already mentioned it in the last slide. In case of prokaryotes, what will be the first amino acid in the polypeptide chain? I will again wait for 2 minutes. Let me know. In case of prokaryotes, the first amino acid in polypeptide chain is which one? Methionine, N-methyl methionine, N-formyl methionine, or all of these? I have already mentioned it in my last slide. Let me know. I will wait for a minute, then I will check the chat box. In prokaryotes, the first amino acids in the polypeptide chain is which one? Let me check the chat box. Let me check if there is any answer. Okay, I don't have any answer, but I have already shown you it in the last PPT. Okay, so okay, let me show you one more time and let us discuss about it. So in case of the prokaryotes, the first amino acid in the polypeptide chain will be, it is number C, N-formyl methionine. Okay, now this is the structure of N-formyl methionine. So what is the structure of methionine? So in case of methionine, just let me draw the structure of methionine. So methionine, we have the structure like this. We have the S here, SME is here, and we have this like, and we have N. Oh, sorry, just I will erase it one more time. Yeah. So we have a carboxylic acid group here, okay, and we have a NH2 here. So this is, so forgive me for the bad handwriting. I hope it is understandable. So this is the S-methyl, and this is the 2 carbon 3 carbon chain, and then we have the NH2 and carboxylic acid at the alpha carbon. So this is the structure of the methionine. So N-formyl methane is nothing but there will be a extra formyl group which is present in case with the methionine in case of the N-formyl methionine. Okay, so that is the difference with the methionine and N-formyl methionine, the structure. So in case of the prokaryotes, although in case of eukaryotes, the methionine is the first amino acid for which it codes for, but in case of prokaryotes, the first amino acid in polypeptide chain is this formylated methionine or N-formyl methionine. So N-formyl methionine also denoted as F-met. If you know, met, MET, met is the short form of the methionine, which is widely used. So in N-formyl methionine, which is denoted as F-met, is a modified form of the amino acid methionine, especially where a formyl group of CHO is attached to the methionine amino group. It plays a crucial role in the initiation of protein synthesis in prokaryotes or bacteria or archaea and organelles of the eukaryotic cells like mitochondria and chloroplasts. So protein synthesis initiation in prokaryotes, protein synthesis begins with the F-met, which is carried to the ribosome by a special transfer transfer RNA or tRNA called initiator tRNA F-met. The AUG codon, which signals for the of the start of the protein sequence, is recognized by the F-met tRNA F-met. The process is facilitated by the initiation factors like IF1, IF2 and IF3 and requires energy for which it gets from the GTP. A specific sequence in the mRNA is Shine Dalgarno sequence which helps align the AUG codon with the ribosome's P site. Post-translation modifications. After translation, the formyl group of F-met is often removed by the deformylase enzyme. The methionine residue itself might also be removed by the aminopeptidase. This modification results in a mature protein with a non-formylated N-terminal methionine or another amino acid if methionine was clipped off. So these are the post-translational modifications which might occur after the synthesis of a particular polypeptide chain. So what is the significance? So F-met acts as a signal marking protein synthesized in the prokaryotes or organelles, differentiating them from those made in the eukaryotic cytoplasm. In some cases, F-met can be involved in the protein degradation pathway, acting as a signal for the protein turnover. The presence or absence of the F-met modification can influence the stability and the function of the proteins. Comparison with eukaryotes, in eukaryotic cells, the initiator tRNA carries an unmodified methionine for the protein synthesis, not F-met. Eukaryotic proteins can undergo N-terminal acylation, which is similar modification to the formylation but with an acyl group instead of formyl group. N-terminal acylation in eukaryotes can also be involved in the protein degradation pathways and can act as a signal for targeting proteins to proteasome or autophagy. In summary, F-met is critical component in the initiation of protein synthesis in prokaryotes and organelles, and its presence and absence can have a significant implications for the protein maturation and degradation. Okay, so this is all about the F for N-formyl methionine, its significance, protein synthesis initiation, how it happens, and also a little bit about the comparing with the eukaryotes and the post-translational modification. Is it all right, or is there any doubt? If there is any doubt, please let me know. Otherwise, I will move toward the next problem. Okay, so I hope it is clear to you. Then I will let me move to the next problem. So problem number 4, it says that what role does messenger RNA plays in the synthesis of proteins? Your options are: number A, it catalyzes the complete process; number B, it provides the genetic blueprint of the protein; number C, it creates a genetic code for a specific amino acid; number D, it modifies messenger RNA molecules prior to the protein synthesis. What role does messenger RNA play in the synthesis of proteins? Again, I will wait for 2 to 3 minutes, and then I will check the chat box. I will for 2 minutes and then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 4. What is the role of the messenger RNA in case of the synthesis of proteins? In the beginning, I have mentioned that the synthesis of protein also is known as the process of the process translation. Transcription is the synthesis of RNA from a DNA, and the synthesis of protein from a RNA, it is known as the translation process. There is also a process which is known as a reverse transcription, where DNA is synthesized from RNA, which is not very common, but it happens. In many cases, it happens. And there is also a process called replication, by which one DNA strand gives rise to the other DNA strands. What role does the messenger RNA play in the synthesis of proteins? Let me know A, B, C, or D, which of the following is the correct answer. I have mentioned already that even if your answer is wrong, it does not matter, but please try. Number A, it catalyzes the complete process. Number B, it provides the genetic blueprint for the protein. Number C, it creates the genetic code for a specific amino acid. Number D, it modifies messenger RNA molecules prior to the protein synthesis. Okay, let me check the chat box. Let me check if there is any answer for problem number 4. Okay, I got two answers, one number C, one number B. Let me check which of the following is the correct answer. Okay, so mm, the correct answer will be, it is number B. It provides the genetic blueprint of the protein. Okay, so the first is like yeah, number C. It's it might seems to be the correct answer, that it creates the genetic code for a specific amino acid, but actually it is not, because the codons already it is present in case of this mRNA. Okay, and only it have the particular tRNA have to read it and bring the particular amino acid. But the more feasible answer or more correct answer will be number B, that it provides the genetic blueprint of the protein. At the beginning of today's session, I have mentioned that central dogma is the flow of the genetic information from a particular DNA to RNA to protein. And now it happens, so how it happens basically, so in case of our DNA, there is a particular gene, a particular space, okay, and which is a course for a particular gene or particular thing, and the RNA polymerase is the enzyme which identifies that particular genetic code and then synthesizes the complementary RNA strand from the DNA. This RNA strand, which acts as a mRNA, and it have this particular genetic code, or particular genetic blueprint of the for the protein, which has to be synthesized. So how it happens, how DNA directs the protein synthesis. So that I will now mention. So these are the steps, okay, of the translation, how DNA directs the particular translation process. So first is the, we have our double strand DNA. Okay, in the cell nucleus. So this is the double strand, double helix structure of the DNA in the cell nucleus. We have the nucleotides. With the nucleotides, I have already mentioned adenine, thymine, guanine, and cytosines are there. Adenine is bound to thymine with 2 hydrogen bond, guanine with cytosine with the 3 hydrogen bond. Okay, and these are the alternative sugar and phosphate groups are present in here. Now, so first is that a step is the messenger RNA or mRNA forming on the DNA strand. So first is the strands of the DNA, they unzip and allow free RNA nucleotides to link with the separated strand. So first is the there will be an unzipping of this particular double helix strand of the DNA, and the free RNA nucleotide, it will start linking on the with the separate strand. Okay, so there will be like here adenine, now adenine will be bond with bind with the uracil, and guanine will be and cytosine, they will bind to each other. But in case of RNA or RNA nucleotides, we do not have the thymine, we have the uracil, right? So the free RNA nucleotide approaches an unzipped DNA molecule to pair its base with the DNA nucleotide, and this is also follows the rules of the base pair. The same thing happens here, and alanine polymerase is the enzyme which catalyzes this whole thing. Then what happens? The mRNA moves from the cell nucleus to the ribosome to the in the cytoplasm, where it acts as a pattern on which amino acids transported by the transfer RNA form a protein. So now, as you can see, these are the free RNA nucleotides, okay, which are joined by the RNA polymerase to form a mRNA molecule. This mRNA molecule, which have this genetic information or genetic blueprint from the DNA, it will go to the cytoplasm and bind with the ribosome, and like this. So this is the structure of ribosome in the first question I have mentioned. So like this, it binds with the ribosome, and ribosome it will now it will then the three nucleotide bases on this particular mRNA, it will act as a codon, and it will code for a particular amino acid. tRNA will bring this particular amino acid, and the formation of the protein chain happens. So this is the formation of the protein on the ribosome. The mRNA moves from the cell nucleus to ribosome to cytoplasm and acts as a pattern on which the amino acid transported by the transfer RNA or tRNA from the protein. Ribosome moves from left to right along the mRNA, and the formation of polypeptide chain. The growing polypeptide, this is the attached amino acid through tRNA. The growing polypeptide chain is this, eventually constitutes the complete protein. Okay, so that is how the particular DNA directed protein synthesis happens. mRNA is the starting point of the translation. DNA is transcribed into the mRNA. mRNA is translated into the amino acid. Each codon corresponds to a particular amino acid like this. You can see here three nucleotide forms a codon which codes for a particular amino acid, like GUA codes for valine, GC it codes for cytosine, AAG it codes for lysine, AGC codes for serine, GCUU codes for leucine. An amino acid form polypeptide chains fold into the proteins. Okay, so that is how the complete protein synthesis happens. So is there any doubt? Let me know. Is it all right, or is there any doubt? If not, I will, if there is no doubt, I will move to the next problem. Okay, okay, you can also let me know your doubts at the end of the session. That won't be any problem. So now I will go to the next problem, problem number 5. Problem number 5 says, which of the following is a stop codon? Your options are like this. Number A, 5 prime AUG 3 prime. Number B, 3 prime AAU 5 prime. Number C, 3 prime UAA 5 prime. Number D, 5 prime GCC 3 prime. Which of the following is a stop codon? I have mentioned what is the codons. I have also discussed what is the start codon. Now the question is related to the stop codon. I will discuss in detail what is a stop codon, how it happens. But first, try to identify which of the following will be a stop codon. Number A, 5 prime AUG 3 prime. Number B, 3 prime AAU 5 prime. Number C, 3 prime UAA 5 prime. And number D, 5 prime GCC 3 prime. Which of the following will be a stop codon? Number A 5 prime, AUG 3 prime, number B 3 prime, AAU 5 prime, number C 3 prime, UAA 5 prime, number D 5 prime, GCC 3 prime. Again, I will wait for 2 minutes, then I will check the chat box. Let me check. I think there will be some answer in the chat box. Okay. Okay, I got two answer. Both are saying this is the number C will be the correct answer. Now let me check if number C is the correct stop codon. So the correct answer will be, it is number B. Okay, 3 prime AAU is the 5 prime is the stop codon. Now let me tell you, first let me tell you what are about the stop codons, and then I will go to why B is the correct answer. So basically this is our coding system. Okay, this is our codon and all the table. This table you will be seeing frequently. So as you can see, there are three marked in blue. As you can see, UAA, UAG and UGA. These are the stop codons. Okay, and methionine, AUG is the start codon, you know. But one thing which you have, I think you have made the mistake in understanding, that the 3 prime and 5 prime. So basically all the codons, all the things, these are mentioned as your, let me just take the pen. So all these things are mentioned as the 5 prime to 3 prime. Okay, 5 prime to 3 prime, all the codons, means in case of these codons also, this will be the 5 prime end, the first one, and the last one will be the 3 prime end. That is how it is mentioned. So if it is if it holds true, then we find that number A and number D are the fine, they are the 5 prime to 3 prime, but this B and C both are written as 3 prime to 5 prime. So that means in the, to be a stop, so what will be the 5 prime to 3 prime? Then for 5 prime it will be 5 prime U A 3 prime, and in case of C it will be 5 prime A U 3 prime. As you can see, 5 prime UAA is, it is a stop codon. Okay, and AAU, you can find AAU, where is this amino acid is present? Yeah, AAU, as you can see, it is not a stop codon. 5 prime 5 prime AAU is codes for the asparagine. Okay, so this is that is why I have given this particular question, because I have seen many students making mistake in this type of question. Okay, because it is a little bit tricky. Not only you have to see what is the codon sequence, but also you have to you have to check if it is given in a particular format, like this 5 prime to 3 prime. Always you have to follow the 5 prime to 3 prime format. All the these codons are given, all these are 5 prime to 3 prime. Okay, that is why your answer, I think you wanted to say that it is UAA is the stop codon, but the codon, the sites you have to think. Okay, because it is this 3 prime UAA 5 prime, it actually denotes 5 prime AAU 3 prime, and it codes for asparagine. It is not a stop codon. The stop codon will be 5 prime UAA 3 prime, 5 prime UAG 3 prime, and 5 prime UGA 3 primes. Okay, these are the stop codons. Okay. So basically, stop codon is a triplet of the nucleotides and mRNA molecule that signals the termination of the protein synthesis or translation. The three standard stop codons are UAA, UAG, and UGA. When a ribosome encounters a stop codon, it triggers a process involving the release factors that halts the protein assembly, ensuring a complete and functional protein is formed. Okay, let me just erase this thing, otherwise it won't be understandable. Yeah. So how it works, the genetic code is set of the genetic code is a set of rules that defines how three nucleotide codons in a DNA and RNA sequence correspond to specific amino acids. Ribosome function during translation. A ribosome moves along the mRNA, reading the codons and recruiting the corresponding tRNA molecules to build a protein chain. Termination signal. When the ribosome reaches to the stop codon, no tRNA with a corresponding anticodon exists to bind to it. Release factors. Instead, specific proteins called release factors bind to stop codon. Protein release. This binding triggers the release of the newly synthesized polypeptide or protein from the ribosome, and the process concludes. Examples and importance: the UAA, UAG and UGA. UAA is also known as the ochre, UAG is known as the amber, and UGA is known as the opal. Okay, so these are the names of this particular stop codons is also mentioned like this. So stop codon read through. Sometimes, due to the errors or the specific context, a ribosome may read through a stop codon and incorporate an amino acid, continuing the translation process. This stop codon read through can lead to a production of proteins that are longer than intended. Understanding the read through is important in the development of therapeutic proteins and addressing genetic disorder caused by the premature stop codons. Okay. So these are all about the stop codon, and this thing you have to remember that all that coding, whatever it is mentioned here, all goes from 5 prime to 3 prime. Okay, so this thing also you have to identify. Okay, so is it clear or I have to repeat it? I think it is clear. Just you have to be careful whenever you are answering this type of question, because most of the time it happens this kind of mistake, so it is fine. Okay, now I will go to the next problem, problem number 6. Problem number 6 says, utilize the chart to identify amino acid sequence given by the following codon series: AUG, AAAA, GGUCU, CAA, AUCG, So this is the codon series. This is the chart. Now you have to identify what will be the amino acid sequence. These are the single letter code for the amino acids. I have mentioned it in my very first class of our this like series of this organic chemistry and biology drug development, the week 1 live session. I have mentioned what are the for one letter course for the particular amino acids, and I have told you to remember, because most of the time questions are asked not from the 3-letter course but from the 1-letter course. So just you have to write it on a copy, the sequence. You have to use this chart, and then you can easily know what is the correct answer, A, B, C or D. I will give you 2 minutes. Try. After 2 minutes, I will check the chat box, and then I will show you the correct answer. Utilize the chart to identify the amino acid sequence given by the following codon series. Utilize the chart to identify the amino acid sequence given by the following codon series. I will wait for another minute, then I will check the chat box. Let me check if there is any answer. Okay, I don't have any answer in the chat box. Okay, let me show you how to do it. This is very simple. Just let me show you. Okay, so what will be the particular polypeptide chain which is denoted by this codon series? The correct answer will be number D. So this is the one letter codes for every amino acid. Alanine is denoted by A, arginine by R, aspartic acid D, asparagine N. Then we have this cysteine C, glutamic acid E, glutamine Q, glycine G, histidine H, isoleucine I, leucine L, lysine is K, methionine is M, phenylalanine is F, proline is P, serine is S, threonine is T, tryptophan is W, tyrosine is Y, and valine is B. So the codon series, as you know every three nucleotide you have to take as a codon. So first we have this AUG followed by AAA, then AGG, then we have UCU, then CAA, then AUC, then GCC. Now the amino acids you have to find from this corresponding chart. AUG is for methionine, met. AAA, AAA is for lysine, as you can see. AGG is for arginine. AGG you have to find here. Yeah, AGG from this arginine. UCU, we will find it here somewhere. Yeah, UCU is for serine. Then CAA you will find it here from glutamine. Then we have AUC for isoleucine, and GCC you will find it here, GCC for alanine. And the one letter code methionine denoted by M, lysine by K, arginine by R, serine by S, glutamine by Q, isoleucine is by I, and alanine is by A. So this will be the polypeptide amino acid sequence of the polypeptide chain which is coded with this codon series. Okay. Now I will go to the next problem. Problem number 7. It says correct order of the steps of the solid phase oligonucleotide synthesis is number 1 deprotection of a DMT group, coupling and deprotection of base. Number B coupling followed by oxidation of phosphorus followed by deprotection of DMT group. Number C deprotection of DMT group followed by coupling, followed by oxidation of phosphorus, and number D deprotection of the DMT group, followed by oxidation of phosphorus, followed by coupling. What will be the correct step order of the steps for the polyphase oligonucleotide synthesis? Number A deprotection of DMT group, coupling, and then deprotection of base. Number B coupling followed by oxidation of phosphorus, then deprotection of the DMT group. Number C deprotection of DMT group, coupling and oxidation of phosphorus. And number D deprotection of DMT group, oxidation of phosphorus and coupling. What will be the correct order of steps for the solid phase oligonucleotide synthesis? The correct order of steps of the solid phase oligonucleotide synthesis will be which one? Let me know in the chat box. Solid phase oligonucleotide synthesis is more or less similar to the solid phase peptide synthesis. I think I have discussed it a little bit in our initial classes, and also I will discuss it in our later of our course in detail. Okay, let me check the chat box. If there are any answer for this problem. Okay, I got two answer. One is saying it is number C, another is saying it is number D. Now let me check which of the following is the correct answer. Number C or number D? Yeah. So the correct answer will be, it is number C. Okay. The protection of the DMT group, coupling, and then oxidation of the phosphorus. So it is the correct answer. So now let me show you. So this is the complete cycle. Okay. The cycle starts from here. So this is the first thing, which is, this is the solid stuff support. So I think I have mentioned in the solid phase peptide synthesis that there is a solid support of mediphyl resin. There is similarly here also is a resin support from which we have the our starting nucleotide. Then for the in this particular nucleotide we have the base in place of this position. The number 2 position we have a base, and number 1, 2, 3, 4, 5, fifth position we have a hydroxyl group which is protected with this DMT protecting group, right? Then first is the deprotection. The acid catalyzed removal of DMT allows the subsequent base addition. So after deprotection of this DMT group, we have this another nucleotide. Okay, this nucleotide has this O is protected with the DMT, and this here we have this on this third position we have a this phosphate linkage type. Okay, now okay. So then what happens? This second nucleotide is added to this position. This number fifth hydroxyl group, which is added like this. The DMT protected phosphoramidate is added. So this is the DMT protected phosphoramidate is added to the unprotected 5' OH of the using a tetrazole activator. This is very important. Okay, so this step is the most crucial step for this, which is the coupling step. A DMT protected phosphoramidate is added to the unprotected 5' hydroxyl group using a tetrazole activator. This is the step 2, which is the coupling step. So first step is the deprotection, second step will be the coupling step. Now the third step is the third and fourth step is the capping and oxidation. So what is this step? So the unreacted 5' are acylated to the prevent the further chain extension. Then the oxidation of the phosphotriester is on the ester to the phosphate using the aqueous iodine. So this is important, oxidation of the phosphotriester to the phosphate using the aqueous iodine. So this is the third step, the oxidation of the phosphorus. So as you can see, this phosphorus is now oxidated, and then what happens is the release and the start of the new cycle. Okay, so this is the oligonucleotide synthesis. That's how it happens, and this is also known as the phosphoramidate method. So what is the importance of the monitoring and purification of the process of coupling efficiency? Monitoring the efficiency of each coupling step is crucial, as even the small failure rates can accumulate and significantly impact the final product's purity. Purification: after synthesis, the oligonucleotide is clipped from the solid support and purified using the techniques like reverse phase HPLC to isolate the desired sequence. Application: genetic testing and research. Oligonucleotides are essential tools for various research applications, including PCR, DNA sequencing, and gene editing. Therapeutic development: synthetic oligonucleotides are used in antisense therapies, gene editing like CRISPR, and personalized medicine for the genetic diseases. And diagnostic tools: they are also utilized in the diagnostic assay for detecting specific DNA, RNA sequences. Okay. So this is the applications and also the importance and monitoring of the purification of this solid phase oligonucleotide synthesis process. So is it clear, or do I need to repeat it? So I think it is clear. Otherwise, please let me know if it is not clear enough, then I will repeat it. If there is no doubt, I will go to the next step. So remember the steps: the reprotection of DMT followed by the coupling, then oxidation of phosphorus. This is also known as the capping. Okay. Okay, now let me go to the next problem. Problem number 8 is says that which of the following statement is true regarding the solid state DNA synthesis? The thing which we have discussed in the last slide only. So number A, starting point is the 5' OH is then attached with the starting point is 5' OH and it is the attached with the silica. Number B, 3' OH is protected with DMT group. Number C, thymine does not need to be protected. Number D, guanine is protected as benzoyl. Which of the following statement is true regarding the solid state DNA synthesis? Number A, starting point is 5' OH and it is attached with silica. Number B, 3 prime OH is protected as DMT group. Number C, thymine does not need any to be protected. And number D, guanine is protected as benzoyl. Which of the following statement is true regarding the solid state DNA synthesis? Which of the following statements is true regarding the solid state DNA synthesis? The starting point is 5 prime OH and it is attached with silica. Number B, 3 prime OH is protected with DMT as a DMT group. Number C, T does not need to be protected. And number D, 1 is protected as benzoyl. I will wait for a minute, then I will check the chat box. Which of the following statements is true regarding the solid state DNA synthesis? Let me check the chat box. Okay, I got one answer which is saying it is number B. Let me check if number B is the correct answer or not. So, the statement which is true regarding the solid phase DNA synthesis will be, it is number C. Thymine does not need to be protected. The starting point is the 5 prime OH and it is attached with silica. It is not true because if you see the start here at the starting point, this 1, 2, 3, this position is attached with silica, but not the 5 prime OH. It is not attached with silica. Okay. And another thing is like the 3 prime OH is protected as DMT group. It is also incorrect because as I have clearly mentioned that this 5 prime OH it is protected with this DMT group. Okay. Now, what is the so this is known as the phosphoramidate method I have mentioned. So this is how what happens. Okay, so this is first we have this CPG bead, then we have a spacer like this, and then we have this initial nucleoside where we have a base 1, then we have this 3 prime position which is attached with the silica, the 3 prime OH is attached with the silica, and then we have this 5 prime OH which is protected by the DMT group. So the first two statements are false because the starting point is not the 5 prime OH, it is the 3 prime OH is this attached with silica, and 5 prime OH is protected as DMT group, not the 3 prime OH. Now here comes the last two statements. So in case of the phosphoramidate method, the amino group of the A, G and C are derived by the addition of benzoyl, isobutyryl and benzoyl groups respectively. Thymine does not have any amino group, so it does not need any protection. This is done to prevent the side reactions. So thymine does not need any protection since it do not have any free NH2 group. So this is the structure of adenine, guanine, thymine and cytosine and thymine. So as you can see, adenine, guanine and cytosine, all these three have the free NH2 group. Like here, in case of adenine, in case of the guanine and in case of the cytosine, we will all have the free NH2 group, but free NH2 group is not there in case of thymine. That is why thymine does not need any kind of protection, but this free NH2 groups of adenine, guanine and cytosines, they are protected. Okay, in the adenine is protected as benzoyl, Guanine is protected as isobutyryl, and this cytosine is derived protected as benzoyl. Okay, why these protections are required? To prevent any kind of side reaction, to minimize any kind of side reaction in case of this oligonucleotide state DNA synthesis. Okay, so there is a special molecule which is attached to a solid support controlled by a pore glass. The initial nucleotide base plus sugar is attached to the special molecule by 3' terminus. The initial nucleoside has been protected by 5' DMT. This dimethoxytretyl, washing with acetonitrile to remove water and any nucleotides, flushing with argon to purge the acetonitrile. So these are the phosphoramidate method. So this the number C was the correct answer. Okay. Now I will go to the next problem. Problem number 9, it says the reaction in DNA replication catalyzed by DNA ligase involves number A, addition of new nucleotides to the leading strand, number B, addition of new nucleotides to the lagging strand, number C, base pairing with the template and newly formed DNA strand, and number D, formation of the phosphodiester bond between 3' OH of one Okazaki fragment and the 5' phosphate of the next lagging strand. Which of the following reaction in DNA replication is catalyzed by the DNA ligase enzyme? Your options are number A, addition of new nucleotides in the leading strand, number B, addition of new nucleotides in the lagging strand, number C, base pairing of the template and newly formed DNA strand, number D, formation of the phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next of the lagging strand. Let me know A, B, C or D which of the following will be the correct answer. I will give you 2 minutes. Read all the options very carefully and then let me know A, B, C or D which of the following will be the correct answer. The reaction in DNA replication catalyzed by DNA ligase involved number A, addition of new nucleotide in the leading strand, number B, addition of new nucleotides in the lagging strand, number C, base pairing of the template and newly formed DNA strand, and number D, formation of phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next on the lagging strand. The reaction in the DNA replication catalyzed by DNA ligase involves which of the following steps? Let me check the chat box. Okay, I got one answer which says it is the number D. Let me check if number D is the correct answer or not. Okay, so the reaction in the DNA replication that is catalyzed by DNA ligase, it will be, yes, it is number D, formation of the phosphodiester bond between 3' OH of one Okazaki fragment and the 5' phosphate of the next on the lagging strand. Okay. So DNA ligase, basically DNA ligase seals the nicks between the Okazaki fragment, converting them to a continuous strand of DNA. It covalently nicks the double strands DNA. Okay. So before going to it, let us understand what is the replication, what is the DNA replications, what are all the steps of the DNA replication. So in case of DNA replication, there are several enzymes that are involved. Okay, and these enzymes play a very crucial role. So first we start with the enzyme helicase that will unwind the parental double helix. So as you can see, look at carefully. So this is the double helix, the parental DNA. One is the 5' to 3', like this dark blue one. Okay, this This dark blue one is the parental DNA. So one goes from 5 prime to 3 prime, another goes from the 3 prime to 5 prime. In case of the first is the helicase enzyme. Helicase unwind the parental double helix. Okay, it forms the replication fork. The single strand stabilizing proteins are there, which stabilize this unwound parental DNA strand. Then we have this 5 prime to 3 prime. So as you can see, this strand. Okay, so this strand is known as the leading strand, the 5 prime to 3 prime strand. So in case of this, so this is the okay. So not it is not the. So the leading strand is synthesized continuously from 5 prime to 3 prime of the DNA polymerase. So now as you can see, since it is 5 prime to is 3 prime is here. So now the DNA strand, the new DNA strand, it will form from the 5 prime to 3 prime in this direction. And this is known as the leading strand because it is synthesized continuously from 5 prime to 3 prime by the DNA polymerase enzyme. So this is very important, the enzyme. So DNA polymerase is the enzyme that continuously synthesizes the DNA from 5 prime to 3 prime. Then there is a strand which is known as the lagging strand. So DNA, for the formation of this particular DNA strand, it is always happens from enzymatically, it always happens 5 prime to 3 prime. In case of the leading strand, it happens continuously, but in case of the lagging strand, it is not continuous. The lagging strand is synthesized discontinuously. Primase synthesizes short RNA primer. As you can see, the red one is the RNA primer, which extended by DNA polymerase to form an Okazaki fragment. So this RNA primer is for with this our primase enzyme with RNA primer and DNA polymerase, it forms the Okazaki fragment. And what does is the Okazaki this fragments do after RNA primer is replaced by DNA by another DNA polymerase. The DNA ligase joins this Okazaki fragment to growing a growing strand. So as you can see, these are also all the Okazaki fragments which are joined by this DNA ligase to form a new strand of DNA from 5 prime to 3 prime, but discontinuously. So basically, overall direction of replication is this. Okay, the 5 prime to 3 prime. In case of the leading strand, it is synthesized continuously. In case of the lagging strand, it is discontinuously. It is produced. So what are the important enzymes that I have already discussed? Like helicase is there. Helicase is unwinds the double helix DNA. We have this DNA polymerase, which basically forms the new DNA strand. Okay, the leading strand, here we have a primase which helps for synthesizing the RNA primer, which is extended by the DNA fragment to form the Okazaki fragment, and DNA ligase is there which joins this Okazaki fragment to synthesize new particular lagging strand of the DNA. And this light blue one is the new strand of DNA, and whereas the dark blue one is the parental DNA. So as you can see, it is a semi-conservative, means there is a one strand is from our old DNA and one is the new DNA. Okay, so this is the thing. So is there any doubt or is it clear? If there is any doubt, you can let me know now. Otherwise, I will move to the next problem. Okay, I hope there is no doubt. So let me go to the next problem. So problem number 10, it says that the position where the tRNA will bind with the ribosome depends on its number A codon sequence, number B intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin like structure. Problem number 10, the position where the tRNA will bind with the ribosome depends on its number A codon sequence, number B intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin like structure. Again, I will wait for 2 minutes. Let me know A, B, C or D, which of the following will be the correct answer. The position where the tRNA will bind with the ribosome depends on it. Codon sequence, intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin-like structure. I will wait for a minute, then I will check the chat box. Let me know till then, A, B, C or D, which of the following you think is the correct answer for problem number 10. position where the tRNA will bind with the ribosome depends on it which structure, or which is the what is the correct answer? Number A, codon sequence. Number B, intramolecular hydrogen bonding. Number C, anticodon sequence. And number D, hairpin-like structure. Let me check the chat box. Okay, I got two answers. Both are saying it is number B. Let me check if number B is the correct answer or not. Okay, the position where the tRNA binds will bind with the ribosome depends on it. It is the correct answer is number C, anticodon sequence. So, so this is the codon anticodon binding of the tRNA. So this is the shape of the tRNA. As you can see, there is a the first is the 3' position where the amino acid binds basically. Then we have a T-Shice arm. There is a variable arm, and there is an anticodon arm is here, and here is the acceptor arm, which is also known as the D arm. So this is the complementary binding. So now this is the codon, which is left for say for example, in case of the our start codon is AUG. So the anticodon sequence must be here. So identify the so for the complementary binding, and then the identification of this codon anticodon binding will prompt the tRNA to take up the amino acid which is coded by this mRNA molecule, right? So what happens? This is the complete thing which is of the translation. I already have mentioned it earlier. Let me just do it one more time. So there is a thing which is called the translational complex. So how this translational complex is formed? Let me highlight the term for you because this is the first time which I have mentioning this term, translational complex. Okay, so in case of the translation, the things that are involved is the tRNA with amino acid. We have a mRNA which have this codons which will code for the particular amino acid. We have a large subunit of the ribosome and a small ribosomal subunit. All this thing comes together to form a particular translational complex, and the tRNA brings the first amino acid in the polypeptide chain to bind to start code for mRNA. So as you can see, this is the anticodon. The codon is AUG, which codes for the amino acid methionine. The anticodon should be this UAC, which is complementary to the AUG, and it will code for the amino acid methionine. So first step is this initiation step where this first amino acid binds. Then is the elongation step. tRNAs bring the amino acids one by one to add on the polypeptide chain like this. Okay, like this polypeptide chains grow from one by one on the tRNA, and then is the termination. Release of the release factor recognizes the stop codon, translational complex dissociates, and completed polypeptide chain is released. So this is the completed polypeptide chain. Release factor, it will identify the stop codon which is UAG. Here also you can see it is 5' to 3' then UAG. Okay, so I have mentioned earlier that how to read this stop codon. So you have to read it like this: 5' to any codon, 5' this particular codon to 3'. So this is the termination step, and then after that components of this translation complex will be recycled to start another cycle for polypeptide synthesis. Okay, so the position where the tRNA will bind to the ribosome, it will depend on the anticodon sequence. Okay. Now I will go to the next problem. Problem number 11, it says the drug that inhibits the attachment of tRNA to mRNA ribosome complex will be number A, ricin, number B, tetracycline, number C, streptomycin, and number D, cyclohexylamine. The drug that inhibits the attachment of tRNA to mRNA ribosome complex will be ricin, tetracycline, streptomycin, cyclohexylamine. Let me know ABC which of the following will be the correct answer for problem number 11. The drug that inhibits the attachment of tRNA to mRNA ribosome complex, number A, ricin, number B, tetracycline, number C streptomycin, and number D cyclohexylamine. What will be the correct answer for problem number 11? Let me know in the chat box. A, B, C or D, which of the following you think is the correct answer? Let me check the chat box. Let me see if there is any answer. Okay, I got one answer which says it's number B. Let me check if number B is the correct answer or not. The correct answer will be yes, it is number B, tetracycline. Tetracycline is the drug that inhibits the attachment of tRNA to the mRNA ribosome complex. So protein synthesis inhibition. So there are several drugs that inhibits this protein synthesis, and these drugs are basically very important in case of the antibacterial in for the as antibacterial therapeutics. So this is our mRNA. We have this tRNA and growing peptide chain. As you can see, this is the 50s portion and this is the 30s. So this is a 70s prokaryotic ribosome. Okay, for the translation. So several drugs are there which inhibit this prokaryotic ribosome prokaryotic translation, and these drugs are basically used as antibacterial drugs. Okay, so chloramphenicol is there which binds to the 50s portion and inhibits the formation of the peptide bond. Okay, then we have tetracycline. This interfere with the attachment of tRNA to mRNA ribosome complex, and we have the streptomycin that changes the shape of the tRNA shape of the 30th portion, causing the code on the mRNA to be read incorrectly. So these are the drugs which actually inhibits this protein synthesis, and these are very crucial in case of because they hinder the bacterial protein synthesis, and they are used as a very potent antibacterial drugs. Chloramphenicol, tetracycline, streptomycin all are used as an antibacterial drug. Okay, like so you have to remember that chloramphenicol binds with the 50th portion, inhibits the formation of peptide bond. Tetracycline interfere with the attachment of the tRNA to mRNA ribosome complex. Streptomycin changes the shape of the 30th portion, causing code on the mRNA to be read incorrectly. So tetracycline block the ribosomal protein synthesis in bacteria. Human mitochondrial ribosome, the 55S are the 39S and 28S. This inhibition by tetracycline is similar. Decreased cytoplasm synthesis, decreased oxphos, and increased aerobic glycolysis and lactic acid. So tetracycline blocks the ribosomal protein synthesis in bacteria like this. Okay, so this here it binds on the 30th on the this here in this position, and interferes attachment of tRNA. So as you can see now tRNA cannot attach with this mRNA ribosome complex. So that is how the tetracycline works. Okay. Now I will go to the next problem. Problem number 12, which says what would be the complementary RNA strand on the following coding DNA sequence in the transcription process. The DNA sequence is 5' GTTACG TGC3'. What would be the complementary RNA strand for the following coding DNA sequence? So take a look, take a note on it that it is a coding DNA sequence which is given. 5' GTTA CGT GC 3'. So your options are number A 5' GUU A CGU GC 3', number B 3' CAA UGC ACG 5', number C 5' CAA UGC ACG 3', number D 3' GUU ACG UGC 5'. So what will be the RNA complementary RNA strand for the following coding DNA sequence, which is given like this in the transcription process. So the process of transcription I have mentioned earlier also, it is the formation of the RNA from the DNA. It is known as the transcription. Translation, formation of the protein from RNA. So first is the transcription, then happens the translation. Now you have to, if it is the coding DNA sequence which is given here, you have to identify what will be the complementary RNA strand of the following coding DNA sequence. I will wait for a minute or two, then I will check the chat box. Let me know then what will be the correct answer. What would be the complementary RNA strand for the following coding DNA sequence in the transcription process? Let me check the chat box. Let me see if there is any answer. Okay, I don't have any answer for this problem. Okay, let me show you how to do or how to identify the correct strand, correct sequence. Okay, the correct answer will be number A. Okay, so in case of our DNA, we have two DNA strands. One goes from 5 prime to 3 prime, another goes from the 3 prime to 5 prime. The DNA strand which goes from 5 prime to 3 prime, it is known as the coding strand or non-template strand or sense strand. Okay, so this is important. The non-template strand or coding strand or sense strand is one which goes from 5 prime to 3 prime. So this term is very important. Let me highlight it. So this non-template or coding strand is the one which goes from 5 prime to 3 prime. Okay. So if the coding strand is given like for 5 prime to 3 prime is this, your 5 prime, so what will be the non-coding or anti-sense or so this is also known as the sense strand. So what is so this is the non-template strand. Okay, so this strand 5 prime to 3 prime, it will not be act as a template for the RNA synthesis. That strand which will act as a template for the RNA synthesis will be its complementary strand, which is the template strand or non-coding strand or antisense strand. Okay, so now let us solve this question. So for example, like we have, so our this is the DNA sequence, this is the coding strand I have mentioned. So this is the coding strand. So if it is a coding strand, this means this is a non-template strand. Okay, so what will be the template strand? Template strand will be the complementary of it. So will be 3 prime, it will be C A will be CAAT G C A C G 5 prime. Okay, so if it is a non-coding strand or if it is a non-template strand, so what will be the template strand? That will be complementary to this coding strand, which will be this one. And now the complementary RNA strand for this template strand, this strand will act as a template for the RNA synthesis, or for it will act as a template for the transcription. So now what will be the RNA strand? That will be complementary to this strand. So we will be now get this 5 prime G. Now A will the complementary of A in RNA, it will be uracil U, because we don't have the thymine here. Then also we have A, then C, then G, then U, then G, C, 3 prime. So this will be the answer. This will be the RNA strand, which is the complementary RNA strand. Here also from image you can see here, okay, that is the mention. As you can see, this is the coding strand or non-template strand which goes from 5 prime to 3 prime. So what will be that? This is non-template. The strand which goes from the 5 prime to 3 prime, it is a non-template. It does not act as a template. That is next one, the complementary one, which is the 3 prime to 5 prime, act as a template strand. So this template strand acts as a template for the RNA formation, and RNA will be the complementary to this basis only. There won't be any thymine, there will be uracil. Okay, so that is how you have to solve this type of questions. Okay, so is it clear? Sometimes the students, they get confused in this question. In last year also there were several students, they did not understand it. So I hope today it is clear. If not, you can let me know. I will repeat it at the end of the session if time permits. Otherwise I can discuss if you have any problem in the next class itself. Okay, so let us go to the next problem. Problem number E says, in the following diagram, E is what? This is the E. You have to identify. E denotes which enzyme. Your options are number A, DNA polymerase, number B, DNA ligase, number C, helicase, number D, lysozyme. E denotes which enzyme? DNA topoisomerase, DNA ligase, helicase, or lysozyme. I will wait for a minute or two, then I will discuss it. Let me know till then which of the following will be the correct answer. In the following diagram, E denotes which enzyme? DNA topoisomerase, DNA ligase, helicase, or lysozyme? Let me check the chat box because we have a couple of questions till now. Okay, I got one answer, number C. Let me check if number C is the correct answer or not. So E denotes which enzyme? The correct answer will be yes, it is helicase. So helicases uses energy from ATP to break the hydrogen bonds holding the base pairs together. This allows the two parental strands of DNA to begin unwinding and forms two replication forks. Each strand of parental DNA has its own helicase. In humans, two inherited diseases, Warner's syndrome and Bloom syndrome, result from helicase defects. E. coli contains at least 6 different helicases, some involved in DNA repair, others in conjugation. The principal helicase in the DNA replication is DNA B. The function unwinding nucleic acids, the primary function of helicases is to unzip double-stranded DNA or RNA by breaking the weak hydrogen bonds between the base pairs. ATP-dependent motor helicases use the energy derived from hydrolysis of ATP or another nucleoside triphosphate to fuel their movement along the nucleic acid strand and separate the strands. Translocation, they are motor protons that move directionally along the single strand, facilitating the separation process. Key process involving helicases, DNA replication. Helicases are essential for separating the DNA double helix into single strands, which are then used as a templates for synthesizing new DNA strands. DNA repair, they play a role in repairing damaged DNA by unwinding it to access the damaged sections. Transcription, helicases unwind DNA to allow the RNA polymerase to access the genetic code. Translation and ribosome biogenesis. In eukaryotes, helicases are involved in the processes such as the translation and the formation of ribosome. Recombination, they also participate in DNA recombination, a process where genetic material is exchanged. Okay. Types of helicases, we have DNA helicases. This enzyme specifically unwind double-strand DNA. We have RNA helicases also. They are involved in remodeling and destabilizing the RNA secondary structures. Significance, helicases are ubiquitous in all known organisms, highlighting their fundamental importance in cellular life. Mutations in helicase gene can lead to the genomic instability are associated with the certain diseases. Okay. So this is all about the helicase, which is a very crucial enzyme in case of the DNA in the central dogma or specifically for the DNA replication and also for the RNA secondary structures stabilization. Now let me go to the next problem. Problem number 14 says which of the following statements regarding DNA synthesis is true. Your options are number A, enzymatically it takes place from 3' to 5' direction only. Number B, enzymatically it takes place from 5' to 3' direction only. Number C, chemically it takes place from 3' to 5' direction only. And number D, chemically it takes place from 5' to 3' direction only. Which of the following statements regarding DNA synthesis is true? Number A, enzymatically it takes place from 3' to 5' direction only. Number B, enzymatically it takes place from 5' to 3' direction only. Number C, chemically it takes place from 3' to 5' direction only. And number D, chemically it takes place from 5' to 3' direction only. Which of the following statements regarding DNA synthesis is true? Let me know in the chat box A, B, C or D which of the following will be the correct answer. Which of the following statements regarding DNA synthesis is true? Let me check the chat box. Okay, I got answer number B. Let me check if number B is the correct answer or not. So the correct answer will be yes, it is number B. Enzymatically, it takes place from 5 prime to 3 prime direction only. So DNA replication, the synthesis of new DNA strands. So DNA polymerase is the enzyme which is responsible with the RNA primer in place. DNA polymerase enzyme catalyzes the synthesis of a new DNA strand in the 5 prime to 3 prime direction. So as you can see, this is the DNA polymerase. These are the nucleotides, and DNA nucleotides, they join all the strands together to form a new strand. Okay. So DNA polymerases are enzymes that synthesize DNA molecule from deoxyribonucleotides, the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single original DNA molecule. During this process, DNA polymerase reads the existing DNA strands to create two new strands that match the existing ones, also performs proofreading and error correction. Properties of DNA replication. The DNA replication is semi-conservative. Each strand serves as a template for the synthesis of new strands. So these are important. I am going to highlight it. So DNA replication is semi-conservative. Each DNA strand serves as a template for the synthesis of the new strand. Okay, producing two new DNA molecules, each with one new strand and the one old strand. This is known as the semi-conservative replication. Replication begins at an origin and usually proceeds bidirectionally. Okay, it begins at origin and usually proceeds bidirectionally. The replication loops always initiate in a unique point, which termed as the origin. 80 base pairs rich sequences are known as the origin sequences. Okay, then DNA synthesis proceeds to 5 to 3 prime direction. Okay, and is semi-continuous. The new strand of always synthesized during the 5 prime to 3 prime direction, with the free 3 prime OH as the point where which the DNA is elongated. Okay. So yes, enzymatically that form takes place from 5 prime to 3 prime direction. Now let me go to the next problem. Problem number 15 says, which of the following amino acids involved in the action of human topoisomerase 2? Your options are number A tyrosine, number B tryptophan, number C serine, and number D proline. Which of the following amino acids is involved in the action of the human topoisomerase 2? Tyrosine, tryptophan, serine, or proline? Last class, I have discussed about the topoisomerase enzyme. I have discussed how it works, what is the mechanism of action of topoisomerase. So I know you can answer it very easily. Which of the following amino acid is involved in the action of human topoisomerase 2? Which of the following amino acids is involved in the action of human topoisomerase 2? Tyrosine, tryptophan, serine, or proline? I will wait for a minute, then I will check the chat box. I already have discussed this question or discussed about the human topoisomerase 2 in last class, so I know you can answer it easily. Let me check the chat box. Hope to find an answer here there. Okay, yeah, I got an answer, number D. Let me check if number D is the correct answer or not. So the amino acid which is involved in the action of human topoisomerase 2 will be It will be tyrosine. It is number A. I have already discussed in my last class. I have already shown you these figures also. So human topoisomerase 2 is a mammalian enzyme that is crucial to the effective replication of DNA. The enzyme binds to the parts of the DNA where two regions of the double helix are in near proximity. The enzyme binds to one of these DNA double helices and tyrosine residues are used to nick both strands of the DNA. This results in temporary covalent bond between enzyme and resulting 5' end to the each strand, thus stabilizing the DNA. The strands are now pulled in the opposite direction to form a gap through which the intact DNA region can be passed. The enzymes then reseals the strands and it differs. So this is the DNA, which are the DNA crossover happens sometimes, and which leads to the very nicked DNA. So what happens? So the tyrosine is there present in the topoisomerase 2, which binds to the DNA strands. In topoisomerase 2, it cuts both the strands, the enzyme, and forms a gap from which the intact DNA can pass. Oh, and that what is the mechanism to which by which the topoisomerase 2 split the DNA change? This is the topoisomerase 2. We have tyrosine. We have a hydroxyl group on tyrosine, which acts as a nucleophile, attacks on the phosphate linkage between this phosphate this phosphodiester bond, and that's how it forms this bond it binds on the DNA strand. Okay, so this is the mechanism. Now let me go to the next problem. Problem number 16 says that colonies of E. coli grown in 15 nitrogen NH4Cl medium is transferred to a medium having 14 NH4Cl as a nitrogen source. The DNA isolated after 2 cycles of growth was subjected to the density gradient centrifugation, and which of the following figures is now the correct presentation? A, B, C or D? Again, I will repeat, the colonies of E. coli grown in the 15 NH4Cl medium are transferred to a medium having 14 NH4Cl as a nitrogen source. The DNA isolated after 2 cycles of growth was subjected to density gradient centrifugation. Which of the following figures show the correct presentation? A, B, C or D? The colonies of E. coli grown in 15 NH4Cl medium are transferred to a medium having 14 NH4Cl as a nitrogen source. The DNA isolated after 2 cycles of growth was subjected to density gradient centrifugation. Which of the following figures show the correct presentation? The colonies of E. coli grown in 15 NH4Cl medium are transferred to medium having 14 NH4Cl as nitrogen source. The DNA isolated after 2 cycles of growth was subjected to density gradient centrifugation. Which of the following will show the correct presentation? A, B, C or D? Let me check the chat box because we have a couple of questions left, so we have to be quick. So I got an answer which says it is number D. Let me check if number D is the correct answer or not. Okay, so the correct answer will be yes, it is number D. So the experiment, this shows that the DNA since replicates semi-conservatively. Okay, so the bacteria grown in this blue one is the heavy nitrogen medium, and this is the light nitrogen medium, the red one. Okay, so the grow bacteria in some heavy nitrogen medium and transfer few to the light nitrogen medium. So what will happen in case of the before the bacterium? So take samples after 0 minutes, 20 minutes, and one round after 40 minutes of 2 rounds of replication. So first from the zero at the first we will get only the parent heavy DNA. Okay, like this. So it will settle down because the nitrogen is a heavy nitrogen, and it will be only heavy nitrogen, heavy nitrogen DNA. Then in the intermediate we will get a heavy nitrogen and light nitrogen, which will have a which is the first generation, and then we will have nitrogen and light nitrogen DNA. Okay, so after two generation, half of the DNA intermediate was intermediate and half was the light only. There was no heavy only DNA. So this is our DNA. This is our semi-conservative DNAs, where we have a parent strand which has a heavy nitrogen and a new strand which has a light nitrogen. And after so from this we will get similar to a this half intermediate, half light, and half heavy, and we will get only the light nitrogen. So this shows this pattern could only be observed if each DNA molecule contains a template strand from the parental strand and the DNA replication is the semi-conservative. So labeling of with isotope E. coli bacteria was first. So this is also known as the Melson-Stull experiment. Okay, E. coli bacteria were first grown in a medium containing a heavy isotope of nitrogen. This resulted in DNA molecules with 15 nitrogen incorporated into their nucleotides, making them heavier than normal DNA. Transferred to a light medium, the bacteria were then transferred to a medium containing a lighter isotope of nitrogen. Subsequent DNA replication would produce a new DNA strand containing 14 nitrogen. Density gradient centrifugation, DNA samples were extracted from the bacteria at different time points after the transfer and subjected to the density gradient centrifugation. This technique separates the molecules based on their density. Heavy DNA, 50 nitrogen, would settle lower in the centrifugation tube, while the light DNA will settle higher. Interpreting the result, after one generation, the DNA formed a single band at an intermediate density, indicating the mix of the 15 and 14 nitrogen, heavy and light, half heavy, half light. This ruled out the conservative replication, where one DNA molecule would be entirely heavy and the other will be entirely light. After two generations, two bands separated. One is the intermediate density and one is the light density. This result is consistent with the semi-conservative replication, where one strand of the DNA forms the original molecule and the other is newly synthesized. The dispersive model, where DNA is mixed throughout, was ruled out. By observing the patterns of DNA density of each generation, Michelson Stahl were able to definitely demonstrate that DNA replication is semi-conservative. This finding was crucial for understanding how genetic information is passed during the cell division. Okay. Now, I will go to the next problem. Problem number 17, it says which of the following is not an inhibitor of DNA polymerase. Your options are number A, cytarabine, number B, gemcitabine, number B, fludarabine, and number D, darunavir. Which of the following is not an inhibitor of DNA polymerase? Number A, cytarabine, number B, gemcitabine, number C, fludarabine, and number D, darunavir. Which of the following is not an inhibitor of DNA polymerase? Which of the following is not an inhibitor of DNA polymerase? Cytarabine, gemcitabine, fludarabine, or darunavir? Which of the following is not an inhibitor of DNA polymerase? Let me check the chat box. Let me check if there is any answer for problem number 17. Okay, there is no answer. Okay, fine. Let me show you what is the correct answer. So, the following, which is not an inhibitor of DNA polymerase, will be number D, darunavir. So, inhibitors of DNA polymerase, the DNA polymerases catalyze the synthesis of DNA using the 4-deoxyribonucleotide building blocks DATP, DGTP, GCTP, and GTTP. The anti-cancer drug cytarabine is an analog of 2'-deoxycytosine and acts as a prodrug. It is phosphorylated in cells to corresponding triphosphate ARA CTP, which acts as a competitive inhibitor. In addition, ARA-CTP can act as a substrate for DNA polymerase and become incorporated into the growing DNA chain. This can lead to the chain termination or prevent the replication of the modified DNA. All of this affects the results in the inhibition of the DNA synthesis and repair. Gemcitabine is an analog of the cytarabine with fewer side effects. The purine analogs of the fludarabine is also metabolized to a triphosphate and has same mechanism of action as cytarabine. It too inhibits the transcription and can be incorporated into the RNA. Now, so these three, the cytarabine, then we have this gemcitabine, and we have this fludarabine. So these are the DNA polymerase inhibitor. But darunavir is not a DNA polymerase inhibitor. Rather, it is a antiretroviral medication to treat the HIV and AIDS. Okay, so it is an antiviral therapeutic. It is a protease inhibitor that works by blocking the crucial HIV enzyme, preventing the virus from maturing and replicating. Darunavir is typically taken with a low dose of another drug, either ritonavir or cobicistat, to increase the darunavir levels in body to improve effectiveness. How it works? It targets the HIV protease. It binds with the active site of HIV protease, an enzyme essential for the virus to process viral protein. Inhibits the viral replication by blocking the protease. Darunavir prevents the cleavage of the viral polyprotein into the functional proteins, leading to the production of the non-infectious viral particle and halting the HIV replication. Okay. So this about the darunavir and antiviral therapeutics we will discuss in our upcoming classes. We will discuss in the due time in our course, which also have this topic. So we will discuss it later. Okay. So now I will go to the next problem. Problem number 18. It says the enzyme responsible for removing the introns and joining the exons together are called number A lysozyme, number B ligase, number C spliceosome, and number D helicase. The enzyme which is responsible for removing the introns and joining the exons together are called number A lysozyme, number B ligase, number C spliceosome, and number D helicase. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 18. The enzyme responsible for removing the introns and joining the exons together is called lysozyme, ligase, spliceosome, or helicase. Let me check the chat box because we don't have much time and I have one or two questions left. Okay, I got one answer which is saying it is number C. Now let me check if number C is the correct answer of problem number 18 or not. So the enzyme which is responsible for removing the introns and joining the exons together is known as, yes, it is spliceosome. So spliceosome is a large and complex molecule formed of RNAs and proteins that regulate the process of the RNA splicing. It is composed of 5 smaller nuclear RNAs and about 80 protein molecules. And the combination of RNAs and these proteins results in formation of an RNA protein complex termed as a small nuclear ribonucleoproteins. These are the mostly confined within the nucleus where they remain associated with the immature pre-RNA transcripts. So these are the known as the in DNA. There are portions which are known as the exons and some are known as the introns. So this exons actually join together to form the mRNA, and spliceosome is the enzyme which is responsible for doing the same. So introns are the untranslated intervening sequences in mRNA. Exons are the translated sequences. This process of RNA splicing, heterogeneous nuclear RNA transcript before the splicing is complete. So introns versus exons. Introns are the DNA, the segments which do not encode any amino acid sequence in the coding region. Exons are the DNA segments which encode a part of an amino acid in sequence of the complete protein. Introns belongs to the non-coding DNA and considered as a basis located between two exons. Incodes belong to the coding DNA considered bases encode an amino acid sequence of protein. Introns are found in eukaryotes, where exons are found in both eukaryotes and prokaryotes. Introns stay in the nucleus by splicing out from the mRNA primary transcript during the mRNA processing inside the nucleus. Exons leave the nucleus to the cytoplasm where production of the mature mRNA happens. Introns found in DNA and mRNA primary transcript and sequences are less conserved, whereas the exons are found in both DNA and mRNA and the sequences are highly conserved. Okay. Now, problem number 19. Which of the following statements regarding RNA is false? Number A, rRNA acts as a catalyst to make the peptide bond. Number B, the amino acid sequence of proteins depends on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes tRNA. Number D, RNA polymerase 2 synthesizes rRNA. Which of the following statements regarding the RNA is false? I will only wait for a minute because we don't have much time. Let me know in chat box if A, B, C or D, which of the following will be the correct answer regarding the RNA. Number A, rRNA acts as a catalyst to make the peptide bond. Number B, the amino acid sequence of proteins depends on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes the tRNA. Number D, RNA polymerase 2 synthesizes the rRNA. Which of the following statement regarding RNA is false? We don't have much time, so let me just check once the chat box and then let us discuss it a little bit. Okay, there is no answer. Okay, I didn't give much time, so it's fine. Let me show you what is the correct answer. So the correct answer will be number D, RNA polymerase 2 synthesizes the rRNA. Okay, so this statement is false. So RNA polymerase are of three different types: RNA polymerase 1, 2 and 3. RNA polymerase 1 is the that only transcripts ribosomal RNA or rRNA. RNA polymerase 2 central enzyme that catalyzes DNA directed mRNA synthesis during the transcription of the protein coding genes, and RNA polymerase 3 is the RNA polymerase that transcripts small untranslated RNA such as tRNA. RNA polymerase 1 transcripts rRNA gene and resides in the nucleus. RNA polymerase 2 transcripts mRNA, miRNA, snRNA and snORNA genes and resides in nucleoplasm. RNA polymerase 3 transcripts tRNA and 5S rRNA genes and resides in nucleoplasm. RNA polymerase 1 transcripts pre-rRNA at 200 copies, and RNA polymerase 2 transcribes 25,000 genes, and RNA polymerase 3 transcripts 30 to 50 genes at a variable copy numbers. RNA polymerase 1 contains the same two non-identical alpha subunit. RNA polymerase 2 has two copies of different alpha-like subunit, and RNA polymerase 3 contains the same two non-identical alpha-like subunits. RNA polymerase 1 is insensitive to alpha amanitin. The polymerase 2 sensitive to 1 microgram per ml alpha amanitin, and RNA polymerase 3 sensitive to 10 microgram per ml alpha amanitin. Okay. So now the last question of today's session, it says the recognition sequence of the Pribnow box -10 is number A 3' TA TA 85', number B 3 prime TTGA CA 5 prime, number C 5 prime TA 83 prime, and number D 5 prime TTGA CA 3 prime. Since we don't have much time, let me just show you the answer, and because we have to discuss little bit about the Pribnow box. So the correct answer is number C 5 prime TA 83 prime. So Pribnow box lies 10 nucleotides from the transcription start point or TSP, and the second was later found in 18 nucleotides away. So the Pribnow box, this is the translation start point. Pribnow box resides in the this TA 5 prime to 3 prime. It is known as the Pribnow box, and there is also, so this is basically on the RNA. Okay. So Pribnow box is a DNA sequence. This is a consensus sequence 5 prime TA80 in the bacterial promoters located at the -10 position, about 10 base pairs before the transcription start site. This functions is crucial binding for the RNA polymerase. Significant, specifically the sigma factor to initiate the transcription, which is the process of copying the DNA into mRNA. The sequence is conserved into its homology, and consensus TA80 sequence determines the strength of the promoter. The consensus sequence, the most common sequence is 5 prime TA 83 prime. It is found in the promoter region of genes, approximately 10 base pairs upstream to the transcription start point, hence known as the -10 box. It serves as a binding site for the sigma factor, which is subunit of bacterial RNA polymerase that directs the enzyme to the correct starting point of the gene. Role in the transcription: by binding to the Pribnow box, RNA polymerase is positioned to begin another process of transcription where the DNA sequence is used as a template to synthesize the messenger RNA or mRNA. Significance: the degree to which the specific promoter sequence matches the TA80 consensus influence the promoter strength, affecting how efficiently RNA polymerase can bind and initiate the transcription. So this is the RNA polymerase. This is the transcription start point. This is the DNA strand, and this is the RNA strand. Okay. So the diagram is a promoter region. RNA polymerase does not copy the promoter sequence into mRNA. We know this is because the promoter is a right to the transcription start point. The transcription start point is a right to the promoter. The RNA polymerase cannot copy promoter sequence. Minus consensus sequence are copied into into DNA, not RNA. Promoter is copied but processed and spliced into mRNA. So the start size is the +1 will be copied to RNA. -10 sequence is the Pribnow box sequence that contained with in all prokaryote promoter region, and -35 sequence we also have a 5 prime TTGSCA is the second recognition site. Alpha subunit recognizes this sequence and connects with it. The RNA polymerase then comes and connect with the Pribnow box. Okay. So this was the last question of today's session. So today what we have done, we have basically discussed about the central dogma of molecular biology, where we discussed about the DNA replication, we discussed about the translation, transcription, and also we discussed about various enzymes that are important in all these cases. Now I am sharing my email ID here. So this is my email ID. So if you have any doubt or any query regarding today's class, please feel free to reach me through the email. Today it was our week 6 live session for this course of organic chemistry in biology and drug development. Our next class will be on next Friday, and it will be our 7th live session on our 7-week course content, which is released today. So please let me know if you have any doubt or any query for today's class or in our any previous classes. I can discuss these things in the upcoming classes. Okay, so I will close the session for today if there is no doubt. Thank you all for joining, and we will meet on the next Friday from the 6 p.m. Okay, I will close the session now. Thank you for joining, and thank you for answering also. If you have any doubt, please let me know through the email.